Institute, University of Florida. And he has his PhD in mathematics, 1996 from University of California. Masters in mathematics and Master of Arts in economics, as well as BS in economics. So this is amazing training George has. So he's also a fellow of Institute of Mathematical Statistics, American Statistical Association, as well as in International Statistics Institute. So thank you so much for coming.
All right. Thanks, Indika, for the invitation. And it's great to be back. And I come back often since I still have my house here. But I pick the right days. It's either a snowstorm or it needs to be very cold. Other than that, I should be out. So this is a really joint project out of what we were doing and we continue to do at MRC2, the Michigan Regional Comprehensive Metabolomics Resource Corps. And so this is joint work with ALA. So in this building, everybody should know ALA. Bill Duran that has worked on the software. Chuck Buran that leads the MRC2. My current student, Jing Ma, I'm going to show you towards the end, some things that we're exploring and they are relevant to the continuation of this project. And Sumanda Basu, my former student in stats, who is right now postdocing at UC Berkeley. And I don't know how many of you have seen Metabolomics. Last time I was in this room, we were having the summer workshop for MRC2. So it's appropriate to talk again about Metabolomics. So I don't know how many of you have seen Metabolomics. And the reason I mention all these people extensively is because through various presentations on Metabolomics, I have freely borrowed from their slides, so they're going to recognize what they have written on various slides in my presentation, starting with Chuck's big picture. So this is, as an introduction, especially in this audience, everybody knows these things. And basically, we're going to talk about data that come from Metabolomics. And given the fact that I don't know how many of you have really handled such data or you know the peculiarities of this data, I'm going to spend a little bit of time, about a few slides, talking about the pipeline so that all of us are sort of on the same page. Probably most of you have dealt with other types of omics data, that they have their own peculiarities. But as you're going to see, once you have pre-processed carefully the data, many of the analysis techniques that you throw at them are very, very similar, and they come from high-dimensional statistics. So again, another borrowed slide. Metabolites are relevant for a number of reasons. Basically, you're interested in metabolic signatures. You may be interested in metabolic flux. Very important, and I'm going to show you some examples of assessing and identifying phenotypes, and so on and so forth. So it's sort of a downstream omics technology, and but it has sort of its own peculiarities. It shares some difficulties that many of you may have encountered dealing with proteomics data, but it has its own special features. So when you do quantitative metabolomics, especially mass spec, you get this type of spectra, and then you're trying sort of to identify the peaks and assign particular metabolites. And then, as I'm going to mention a few slides down the road, it depends on some peaks you're able to identify, some others you're not able, and basically that's what this talk is all about. Whether we can leverage some recent developments in modern high-dimensional statistics to help us going from the peaks or the features that we have identified and leverage that information to improve our identification of unknown peaks. And this is a big issue here, and this is one particular approach. And basically, I'm going to walk you through the methodology of how you can do that. So as I've mentioned, just for all of us to see what's going on with this type of data, this is a very typical data processing pipeline. As usual with all the omics technologies, you start with quality assessment, quality control type of issues. And in one type of mode of operation that's coming up, what is known as the untargeted approach or unbiased approach, you end up having a lot of missing data. Many of these peaks you cannot really detect them for particular samples. So imputation becomes an important issue. as with all the other omics type of data that you get, you need to worry about normalization, removing batch effects, and so on and so forth. And then you start getting into similar types of analysis that many of you are familiar, whether you're dealing with microarray data or proteomics data, you know, exploratory analysis to see what's going on, both from the perspective of your metabolites or features and from the perspective of your samples. Then usually you don't go on into differential analysis, especially if you have 2 or more groups or biological conditions, or constructing of classificatory models, or associating them with an outcome variable. And at the end of the day, you know, if you start sort of taking a more systems perspective, you would like to get into pathway enrichment analysis. And a number of things that I'm going to talk about, roughly speaking, are related to network analysis. In this particular case, we're going to use particular network models that you can construct from metabolomics data in order to help us identify these unknown features that you have in the spectra. But towards the end, I'm going to give you sort of a preview, just a couple of slides, of where you can go beyond pathway enrichment analysis and how you can start thinking about networks across different conditions. And people have talked in the literature about differential network analysis. So you're going to see a sneak peek of where we're going with this project. So one thing that you have, you know, when you process your pipeline of metabolomics data, so basically, it's not only that you process your samples, but in many cases, we put these pooled samples in order to control or to be able to say something about the quality of the run and see if everything was correct. So basically, what I'm showing you here is sort of the distribution across all the metabolites or all the features that you have collected for all the samples. And these were sort of the pool samples. And that tells you about the quality of the data that you may need later on for some of the tuning parameters that I'm going to talk about. And this was the baseline, and these were sort of the experimental conditions. So the advantage is that in metabolomics and similar in other mass spec technologies, you have this control type of samples in order to assess the quality of your analysis. And this is sort of familiar with other omics data. You need sort of to normalize them appropriately before any downstream analysis. Exploratory analysis depends on what you are interested in. Principal component analysis, a popular technique. Cluster analysis, where you're trying sort of to do joint clustering or bi-clustering of both your samples and potentially selected features, and see whether sort of you have signal. Basically, these are sort of techniques, multivariate analysis techniques, as we will call them in statistics, you know, to get the big picture of what's going on, both in terms of your samples and also in terms of your variables or features. Then the usual differential analysis. Pick your favorite t-test under whatever conditions you would like to assume. And what type of testing procedure you use depends on how you have normalized the data, or another way sort of to start building classificatory models to separate normals from disease state or treatments for controls and so forth. And this shows, you know, variable importance plots for random forests. I mean, all these are secondary to my talk, but basically I'm just trying to give you the big picture of what happens with metabolomics data. So as you've probably noticed already, you know, there are some particular issues, especially with the quality control and how you assess the quality of your run. And, you know, once you have normalized the data, basically, these are very common techniques that many of you have used when you deal with microarray data or RNA-seq data and so on and so forth. At some point, you just have, from a statistical perspective, high-dimensional data, few samples, lots of features or lots of variables, and then you start using your favorite technique, and depending on what question you're trying to answer. So there are lots of similarities once you have processed the data, the omics data, of how you proceed downstream. But at the end of the day, in most cases, what you would like is sort of to start getting biological insight, and that's where this business of pathway mapping and enrichment analysis comes in. What I'm showing you here is different networks that we can construct from the Medscape tool that Ala has developed and continues working on it, and I'm going to show you how we have started integrating the new methods in the new version of the tool so that all the technology and the tools that we're developing can be sort of seamlessly used. And pathway mapping and enrichment is important because you start taking, as I've already pointed out, a more systems perspective point of view. So right now I'm slowly moving to the sort of the core of the talk and what we have done. One thing to keep in mind is that when you're doing metabolomics analysis, depends on what mode you operate. If you're doing a targeted analysis, basically, you're using an assay where all the compounds that you're interested in, you're going to identify, you're going to have obtained data. Usually you don't have missing data. So, but the downside is that basically you're going to have data for those compounds that are in your assay. All the previous things that I've talked about, normalization and then downstream analysis still hold, but the big advantage if you're in a targeted mode is that you have complete data or nearly complete data. From experience, the imputation that you need to do is very light and it's almost inconsequential. On the other hand, if you know what you're really after, a targeted analysis, and provided that such an assay exists, it works fine, and that's about it. On the other hand, if you would like sort of to cast a wide net and basically be in a global profiling mode, then usually you're going to do an untargeted analysis, and that's where a whole bunch of issues come up, and that's where we're trying sort of to use some of the methods that I'm going to describe in a second to help identify a lot of unnamed features. So in the untargeted mode, what are the challenges? First of all, you have a lot of missing data. So the first thing is that you detect you have a lot of features. You have detected a lot of peaks in your spectrum, and only a small percentage, as I say on this bullet point, only a small proportion corresponds to identified metabolites. So here depends on how you chose your cutoff because you had a lot of missing data. So here you're asking for 70% presence in your samples of every feature, then you're left with about 4,000 features. If you make your cutoff more stringent, you start cutting down on the number of features. But even if you ask that feature was present in all your samples, you still have thousands of features. And many of them, and the majority of those, are usually unnamed. And therefore, for some types of analysis, if you're trying sort of to see if you have signal to differentiate your two groups, let's say treatment and control or normal and disease, you don't really need to know the features because all the techniques that I mentioned in the previous pipeline, many of these exploratory or other techniques like building classificatory models, the only thing that they need is these features. And then they're going to select which ones are more capable of separating the two groups and so forth. But if you start trying to do this business, and therefore do a more informed analysis and try to understand from a biological perspective what is going on, somehow you need sort of to identify your features in order to be able to start populating these pathways and these networks. So that's where this is the big challenge because think of usually about 90% of your features are going to be unnamed, and therefore the question is what you do. I mean, Alex, what he's going to tell you is you need to work very hard and in order to start identifying features. And what I'm going to present is, basically built of some ideas of how you can correlate unnamed and named features if you have those, and especially if you have data both from targeted and untargeted analysis, how you can sort of try to integrate that information and provide good hypothesis of what these features may be. And of course, you still need to do sort of the validation, but basically that's where we're going. So that's why this is sort of an important topic, because in this type of analysis, which is very popular, you end up having all these features, and then you can do some types of analysis, but there are a number of other questions that you cannot really answer, and especially if you're really interested in pathways. So basically, more or less, I have covered the first bullet point. And where I'm going, next is the proposed approach is going to leverage correlations, both classical correlations, and I don't know how many of you have seen the notion of a partial correlation and the corresponding partial correlation networks, primarily between named metabolites that come from an untargeted analysis, or in many cases, if you have appropriately normalized your data, you can pick those from a targeted assay. And unfortunately, I mean, in terms of correlation networks, that's easy to do. But unfortunately, when you start building these partial correlation networks, there are some technical challenges. Basically, the key aspect is that the number of features, as we've seen, usually far exceeds the number of samples available. I mean, even if you have a large study, you may have 200, 300, 400 samples. But as we have seen here, even with a very stringent cutoff, basically we're looking at thousands of features. So when you're trying to do this partial correlation business, basically you start running into technical problems. And I'm going to sort of describe how we have addressed some of these issues. OK, so this is sort of stats 101. So this is sort of the definition of Pearson correlation. Many of you have used that. And so if we have P metabolites, and basically we have measurement on N samples, and at this point I'm assuming that I have imputed the data, have normalized the data, so everything is nice. Basically you can calculate the correlation coefficient between any pair of those. So we have P squared order of correlations. And then in that case, basically we can build a correlation network, and many of you have done that on some software platform. They are very easy to calculate, but essentially they capture co-expression between two metabolites or two features, if they are unidentified. And from a statistical perspective, they capture marginal associations. And therefore, they don't sort of distinguish between direct versus indirect connections. So in order sort of to be able to delineate between these two types of connections, statisticians have thought of another way of measuring correlations, which is somewhat more involved. So this is the notion of a partial correlation. And basically what we are trying to do is, instead of just looking at the co-expression between two metabolites, basically this is what the covariance captures, and then you just normalize it. That's the whole idea of correlation. Basically, the covariance captures this association. Here, you're trying to capture this association, but conditional on the effects of all the other features in your data. And one way sort of doing it is basically you use the measurements on one metabolite as your outcome variable, and then you regress it. This is a standard regression model on all the other variables. And basically, you calculate, once you have calculated all these regression coefficients, then you get the residuals. Then you go on to the second metabolite, and you do exactly the same exercise, and then you get the residuals. Basically, your data minus the fitted values, this gives you the residuals. And then you calculate a Pearson correlation on the residuals. So basically, what you're doing is you account for the effect of all the other metabolites in your data. So that's the key idea behind partial correlation. And the key aspect that, what this sort of gives you is, suppose that you had a metabolite that was driving the associations of two other ones. So basically, this was sort of the key driver node in a particular condition. So if you were to look, given the fact that both these other two metabolites are influenced by this third one, when you did this type of correlation, what you're going to see is very strong correlations on all three pairwise entities. Because even this third one, given the fact that it's impacted by these other two, is going to show you a very strong Pearson correlation. On the other hand, when basically you do this type of exercise, you're going to identify that this was the driver node. And basically, this starts allowing you to identify direct versus indirect association. And that's the idea behind marginal associations and conditional associations. As you can see, from a technical perspective, this becomes a more involved computation because then you need to do all these regressions. And the other thing that you need to keep in mind, given the fact that you have these regressions that you need to perform, this is going to work if you have more samples than metabolites. So going back to what we were discussing, especially in untargeted studies, you see where we're going. We already start sensing that there is going to be a problem because usually you have 2,000 or more features, even after thresholding and throwing those that have missing data, and we are not going to have 2,000 samples. That's, you should be thrilled if you can never measure 2,000 samples, at least in 2015. Maybe in a few years, that's a different story. So, okay, so let me switch gears a little bit and connect all these things, assuming that my data come from a normal distribution. Yeah. So right now, the assumption that I make is that my P metabolites follow a multivariate normal, and all of you have seen sort of the picture, or in statistics courses, we have tortured you with a bell-shaped curve, the picture of a normal distribution, and in high dimensions, basically you have sort of this multidimensional bell-shaped curve. So, and let me introduce and call omega the inverse of the covariance matrix. So as you remember, the key idea when we were calculating Pearson's correlations was basically calculating the covariance and then normalizing it. So basically, these marginal associations come directly from the covariance. Basically, that's what it says. So two of metabolites are marginally independent if the corresponding correlation is 0, which is equivalent of their covariance to be 0. On the other hand, if you're doing partial correlations, basically you are looking at conditional independence because basically you condition on everybody else. This was exactly the definition that we had. And the nice thing is that if you make this normal assumption, and how well justified this is in your data depends on how you have normalized the data. So if you feel fairly comfortable that this assumption roughly holds in your data, then it's easy sort of to calculate in one shot both Pearson and partial correlations, and therefore then put them in a network. So in that case, the network, the edges on the network, they're going to correspond either to your correlations in the correlation network, and therefore the strength of the edge or the weight of the edge is going to capture the strength of the marginal association, or if you're calculating a partial correlation network, the weight of the edge is going to correspond to the partial correlation. And computationally, under this assumption, things become easy because either you're calculating the covariance matrix, and those that are marginally independent just correspond to entries in your covariance that are 0, or you're taking its inverse, and whenever you find 0 entries, then basically correspond to metabolites that are conditionally independent. So in my little sort of toy example, accounting for this driver node, these two are conditionally independent, and therefore there shouldn't be an edge in the corresponding partial correlation network. Okay, so this is fine, but as you sense, this is going to start becoming a critical assumption because if you have more features, more metabolites, or more features than samples, then your covariance matrix is not what we call in math full rank, and therefore is not invertible. So somewhere this framework starts breaking down, and therefore we need to do something, because otherwise, in the type of problems that we have, where you're always going to have, at least at present, more features than samples, you're never going to be able to calculate this partial correlation. Okay, so moving on. So this is sort of the general technical framework. There is more technical stuff coming up, but basically this gives you what Pearson's correlations capture, what partial correlations capture, and how you can calculate them under the normality assumption of your data. But the way we're going to proceed is we're only going to focus on So the problem is that even if you try to use sort of this machinery, basically if you have 2,000 features, basically you're trying to construct a humongous network that has basically 2,000 by 2,000 entries, even if you had enough samples. And basically that means that any association, any conditional association in the partial correlation network that is not zero is going to be present. But some of them may be very, very weak, which means that, yes, you are picking them up, but from a biological perspective, they are not giving you really any information. So, so here comes the point where Chuck had a very critical insight. And basically, the next slides, I'm trying sort of to translate or to justify technically within this framework Chuck's insight. Basically, Chuck kept saying that, well, when I look at the correlation networks, Pearson correlation networks, I would like to threshold them and only concentrate on that part of the correlations that are large, either positive large values or negative large values. But I don't really want to look at correlation coefficients that are close to zero. So basically, our strategy is going to be that we're going to first calculate the correlation network that you can always calculate, because you're not suffering from this issue. And then only focus, basically, we're going to only screen the strong connections. Basically, we're only looking for associations, marginal associations between metabolites that have large positive or negative correlation. And then we're only going to sort of look at that subset of named and unnamed features. Then eventually, we're going to do a little bit more screening and calculate the corresponding partial correlation network to identify the key driver connections, as I explained. So the question then is, I mean, if you calculate everything, then you know exactly how the correlation coefficients and the partial correlations coefficients are connected under the normality assumption through this mathematical relationship. But when you start sort of just screening first, basically you're looking at a particular subset of your data, and then the question is, and this is exactly what we would like to do in order to further reduce the data, but then what is the connection then between the Pearson and partial correlation networks and the screening? Basically, that's what I'm asking. So I'm not going to sort of bother with too many technical details, but basically, these are sort of two key ideas that come from what we call graphical modeling, and especially for Gaussian graphical models. And basically, I'm trying sort of to show you the key idea in terms of these pictures. So we are going to have that these metabolites are deseparated, given the remaining ones, if basically these remaining ones block any flow of influence from N1 to N2. And basically, this is sort of the picture. So these remaining ones create this blocking type of structure. And then there is another key idea, basically, that comes again from this graphical modeling. And those of you that have seen direct acyclic graphs and distributions over direct acyclic graphs probably have seen the idea of faithfulness of a distribution over a direct acyclic graph. Basically, roughly translating in our setting is if the total effect from metabolite 1 and 2 and from 2 and 3 does not cancel the effect of 1 to 3. So basically, by using sort of and putting these two things together, under the faithfulness assumption that depends on this idea of this separation, the features that have high partial correlations also need to exhibit strong marginal correlations. So, the math gets a little bit more complicated, but basically this bullet point is the key idea for operationalizing Chuck's strategy of first, screening in terms of marginal correlations, and then using all of those features that have strong marginal correlations and go into partial correlations. Okay. So, of course, in our data, we need to be a little bit more careful, because when we calculate the partial correlation network, we are going to have components that involve only unnamed metabolites. And in that case, it's very difficult to see, what happens next. So basically, the way we're going to sort of proceed, at least now, is we would like sort of to find strong marginal correlations, Pearson correlations, between at least one named metabolite and a number of unnamed features. So we're going to just start sort of concentrating even further. And we call these blocks, these seeds. And basically, we're going to construct partial correlation networks among all the seeds that we have identified that are connected to unnamed metabolites. And given the fact that you have thousands of unnamed metabolites, even after all this screening, you still have estimating large partial correlation networks. Okay. So I hope that the strategy is clear. Basically, we would like to calculate both the marginal correlations, the marginal associations with the Pearson correlation and the partial correlations that give us this conditional dependence or independence relationship between our metabolites after a screening step. And the previous slide justified, and this bullet point justified why this is sort of kosher to do within this framework. So basically here I'm describing the algorithm of how we can construct a partial correlation network. And the key aspect is that we use a pre-specified threshold to screen out lowly correlated pairs of metabolites. And you can pick your threshold 0.5, 0.6, 0.7, depends on how much you would like to screen out. And then, of course, after we have done the screening, you still would like to see, depends on how much data you have, which ones may still be sort of 0 or different than 0. And in that case, basically, you run a test, you get the p-value, and of course, given the fact that you're testing lots of correlations, you need sort of to adjust for multiple comparisons. So I'm going to come back and discuss this data set in more detail. But even after all this screening, this is the picture that we get from this paper, you still have kind of a hairball. So how do we construct a partial correlation network even after our screening test, assuming that we have more samples than metabolites of features? So this is sort of still a straightforward procedure. So again, we are going to use sort of a threshold, and we are going to use one of the procedures that I described before, this regression type of procedure. And then we're going to again run a test to see if our partial correlation between two metabolites is 0 or non-zero. And again, we're going to adjust the corresponding p-values for multiple comparisons. And this is sort of a picture that comes, again, borrowed from this paper. And what you see is, and that's what you expect, but this is a much more sparse network, and you can start seeing, what happens. Remember that the edges here are capturing conditional dependence relationships, statistical relationships, because you have conditions. When you look at this edge, you have condition on the information from all the other nodes in your network. Whereas here, these are just marginal associations. And this is sort of the interpretation of an edge in this context. Okay. So the question then is, what happens in the setting that we're going to be in our studies, where the number of samples is significantly smaller than the number of features, even after the screening test? So basically, as I've already alluded to, these operations start breaking down because they require n to be larger than p. So over the last 5, 6, 7 years, in the statistical and machine learning literature, there has been an enormous amount of work of what happens in these settings. And of course, one key assumption is you assume that the inverse of your covariance matrix that you're looking at is sparse. So basically, you assume that there are not too many connections. So this is sort of an assumption that you need to make if you would like to make progress. And in general, I mean, these type of networks are somewhat sparse. How sparse, that's sort of a different story. But basically, you need to make such an assumption. And in that case, basically, there is a lot of machinery of how you can estimate your partial correlation network, even if when the number of samples are fewer than a number of features. And this is a famous paper that basically solves the corresponding constraint optimization problem. And for those that have seen sort of this type of framework, given the fact that you're calculating an inverse of the covariance matrix, you need to make sure that your final estimate is positive definite. And this is exactly the constraints that you need to carefully deal with. So basically, the data that we have after normalization, and we have long transformed and centered and scaled appropriately, we have the metabolites on this dimension and the samples on this dimension. And keep in mind that this is a short and fat matrix because we have many more features of metabolites. So this is, and we have significantly fewer samples. So this is a really fat matrix and short. And this is the mathematical problem that you solve. And basically, this enforces this L1 constraint. And at the same time, you still need to operate in the positive definite cone of symmetric matrices. This is sort of the other constraint. So a few comments. This is by now very well understood technology. There is great code in R or in many other packages, in other languages. So people, you can run it very routinely. It's fast, it's easy to implement. It provides a good estimate of the sparsity level, even if you have fewer samples than metabolites. One issue that is still fairly open in the literature, and there are lots of suggestions, is how you tune this parameter, because basically this is the parameter that enforces the sparsity. So basically, it makes all these things work. And so this is still a challenging issue. From a technical perspective, what we know by now is that these estimates are generally biased. So that's something that you may be concerned about. And also, you don't have any uncertainty associated with the estimate. In this context, you don't get distributions about your estimates, and therefore, you cannot construct confidence intervals. Or until recently, you couldn't construct confidence intervals. So basically, this strategy that was working well in this context, because basically you could run a hypothesis test and you can construct either a confidence interval or get a p-value, that breaks down. You get estimates, but you don't have a measure of uncertainty. So, but especially one key issue that people have noticed, both theoretically and in a lot of empirical work, is that your estimates are biased. And people have worked really hard to start resolving some of these issues. And this is very, very recent work. It's not ours. We're borrowing from the literature. And Van de Geer is one of the top theoreticians in the field. So, and basically, she proposed with her student a procedure both to correct the bias that we're getting, and also for getting asymptotic confidence intervals for your partial correlation. Basically, based on this work, suddenly you have a lot of goodies that you can leverage on. And this is sort of the algorithm that you get. And the key aspect is that basically you get, in an appropriate normalized form, you get uncertainty assessments for your partial correlations, even when the number of samples is smaller than the number of features. So this is sort of a great recent development that we leverage it in order basically to estimate our partial correlation network. So let's start looking at some results and what happens. And always keep in mind that the key aspect in this business is, you know, how you use this tuning parameter. Basically, if this tuning parameter becomes too strong, you estimate a very sparse network. Basically, you are asking to estimate very few partial correlations. If you don't tune it very strongly, here you start getting many, many more of those. And this is sort of the intuition behind how this parameter works. And there are suggestions in the literature. Basically, you see asymptotically, people have calculated the order of these parameters, but you know, in finite samples that we operate, you need to do much more work in order to really tune it. And the procedure is very sensitive to how you pick these tuning parameters. And as I said, it's still a challenging issue. Okay, so let me walk you through this sort of gold standard data set that came from a German group. And this is, you know, something that rarely are you going to have access to such data. Because basically what they did was you have an enormous sample size. You have 1,000 plus samples, and they are looking at very few metabolites, 150 of those. So basically, we're in a very nice regime where your number of samples is much larger than the number of features. So you can do lots of things. So when we calculate the Pearson correlation and we use the very stringent adjustment of the p-values, we get a correlation network with about 5,500 edges. The total number of edges that could have been present is 150 square, and therefore it still has a certain level of sparsity. Not all metabolites are correlated with each other, basically, that's the upshot. When you do the partial correlation network that you expect to be much more sparse, again, under the same correction for multiple comparisons, which is very stringent, basically you only get 405 edges. And basically these were these two pictures. Here you still have 5,500 edges and you get this hairball type of network. Here you have only 405. And basically here you can start identifying the key driver nodes, going back to what I was saying at the beginning of my talk. So given the fact that in this context we have lots of samples, and therefore basically you can invert the covariance matrix and calculate everything. Here we haven't done any screening. The first experiment is to assess what happens when you don't have that many samples. And the number of samples starts becoming of the order of your number of metabolites to see what's the sensitivity of our procedure and how well we tune it. So basically we start, the first numerical experiment that we undertook was, this is the complete data set, then we cut it to half, then we cut it to half, and then basically we took a value that is very close to the number of metabolites that were present, and then another value that is, you have, in this context, you have fewer samples than metabolites, which is gonna be exactly where you're gonna operate with your own study. We fit the model the way we describe it, both by just calculating the inverse of the covariance matrix and then using this debiased Lasso procedure. And basically, we are trying to look at how well we recover the edges compared to what we had when we had a lot of samples. So when you start doing this upsampling, how much do you lose? Because basically, you don't have as much information as when you have 1,000 samples. And then the other performance metric that we report is, well, some of these edges or some of these correlations or partial correlations are not as strong. So if we only are interested in the strongest partial correlations, how many of those do we recover? So the upshot, and I'm going to show you the table on the next slide, is that if you use a lot of samples, 500, of course 1,000, both procedures perform well, and therefore, basically, the key message for us here was that we know how to tune our procedure, because that's the key aspect. When you start getting into these settings where the number of samples is about the same as the number of metabolites, or in such a setting where you cannot invert the covariance matrix anymore, our procedure still performs well, or relatively well. So basically, here are sort of the results. So if when you use all the samples, and under this stringent correction for multiple comparisons, you get the network of 405 edges, and the strongest 10% edges in terms of magnitude are 41. So then, if we used our procedure, we estimate a few more compared to this, and we have agreement of 62%. So basically, we start having some false positives. And basically, we recover all the top edges, all the strongest edges. Things are, okay, this is kind of boring because you could have used this procedure, so you didn't really need all this machinery. Here is where things start getting more interesting, because here you start having your sample size, and our recovery is still pretty good, and we play the same game with the other procedure, assuming that this is sort of the truth. So in this case, when you have your number of samples, basically you recover correctly 50% of these 405 edges, but basically you recover essentially all the strong ones. When you go to the next level, again, here you still have 500 samples and only 150 metabolites, which you're not going to have in most real studies. Here you start getting into the regime where the number of samples is about the same as the number of metabolites. Our procedure, at least in terms of the strong ones, is going strong. By trying to invert the covariance matrix, it starts breaking down. Here we are basically, your n is equal to p. This is crap. Here we're still going fairly well. And we still have recovery of the strongest edges. Here we have fewer samples than metabolites. So basically, the two things that we need to keep in mind is that we know how to tune the procedure, and therefore, at least as long as you're interested in the strongest edges in your network, you're going to recover most of them fairly correctly. And we have a procedure that right now we can unleash on real studies. So here are sort of the pictures of how the networks look when we had all the samples, and there are about 405 correct edges. And here are how the two procedures perform. Basically here, you get very little if you're trying to invert the correlation metrics, but our procedure still works well. So let's do on another study. These are data from chat. Well, the problem then is that, of course, you can use the Penrose inverse, but then, you know, what are exactly calculating in terms of the partial correlation? A generalized inverse is not exactly like the inverse, and therefore, I'm not quite sure how to interpret it. I mean, all the things that come under this Gaussian graphical modeling, yes, I can calculate some sort of an inverse, but has very different properties than what I'm looking for, and the statistical interpretation. Because here I'm looking for these conditional dependencies. So mathematically, yes, I can take an inverse, but I have lost all the other things that I would like to do. Okay, so this is a sample of, so here we have essentially 240 samples. Of course, there is dependence between these samples because these are 120 women on 2 consecutive visits. So here, when we tune the model, we need to be careful to take that dependence into consideration. And exactly how, when you have dependent samples and how you do all this business, that was part of Sumanta Basu's dissertation. And after we have sort of cleaned the data, we have 3,000 features. So right now we are in a challenging setup. And also we had some targeted data. So we ran an untargeted analysis and we get 3,000 features. And then we also have some additional metabolites from a targeted assay. And of course, most of these are going to be somewhere there, but in many cases, you may not identify all of those. So I'm going to show you some snapshots of what comes out of all this methodology. One thing that we've noticed is that basically you start out of these features, basically you have in-source fragments. So basically the metabolite has fragmented. And basically this corresponds, all these fragments correspond to valine and this to proline. And therefore this is very helpful to go back and start cleaning the data set. Alex has other thoughts of how you can do that, but this is, and there are other ideas out there, but this is one way that coupled with ideas out in the literature can start helping you identify all these in-source fragments and therefore go back and start cleaning your features. So that's one outcome that you get out of all this. The other outcome that you can get is basically you had a number of amino acids from the targeted assay, and then using these seeds and partial correlation network, you can start populating an amino acid network. And this is going to be helpful when you start thinking about pathways and enrichment and so on and so forth. So right now here, we'll start talking about taking a more systems perspective. And of course, the last thing is sort of validation, because here by doing the seed and the unknowns, so basically here with a lot of additional work, you start identifying the unknowns, and that was sort of the key question that we asked at the beginning of the talk. Of course, this still requires a lot of manual labor, because once you start looking at all these subnetworks, in order to really identify what are the nodes, it requires quite a lot of work, but basically that's where the framework, or we're going with this framework. So basically, these are the three possible uses that we have identified. This is to clean up the data set in terms of the features. This is sort of to start populating particular networks that we're interested in, and this is sort of the killer application of sort of using all this technology to identify these unnamed features. These are slides from Bill that has worked on integrating all this into Medscape. So this is sort of new developments on the Medscape platform. You input your data, you select your parameter, you select your normalization, you select your parameters, you select your thresholding, and then you run the partial correlation. So this is sort of the input file. And then using the slider, you can decide how much you would like to select on the Pearson correlation network before you start looking at the partial correlations. Here are some screenshots. And then basically, you start visualizing the result and start zooming in order sort of to understand what goes on. A little bit of additional background on Netscape. It leverages KEGG and can do all these nice things. I'm pretty sure many of you have heard Ala giving a much more in-depth presentation about her tool. So the last thing that I would like sort of to, before closing, is this sort of the summary. Basically, we used all these recent developments in high-dimensional statistics of how to estimate partial correlation networks, even if you have fewer samples than features. And then, we're building, Ala's team is building sort of the extensions to the Medscape platform so that everything works seamlessly. And of course, then the key question is, how to sort of prioritize all the things that have you come out of this analysis, because you still have hundreds of potential targets. So which one should you really focus to validate? We have a number of ideas of how you can extend it to lipids. One issue is that still the method requires considerable sample sizes. So one question then is, there are so many metabolomics data sets out there in the public. Other labs have run similar studies. How can you start leveraging all that information in order sort of to build such networks? And the last thing that I would like sort of to mention is, when you estimate the partial correlation network, you can sort of start using additional constraints that you may think are useful, like chemical similarity, which helps you sort of constrain the search space. So this is from a technical perspective, can easily be incorporated into the methodology. And the other thing that you may want to do is, in all this data that I showed you, there might be two different biological conditions. And therefore, what you may start asking is, why shall I sort of just estimate a single network, both of named and unnamed features? There might be differences in the two networks. But the assumption that I make is that there are some common or similar components. So when you start thinking along those lines, the question is how you do sort of this joint estimation. And that's basically the piece of my other PhD student that is working on the project, Jing Ma. And these are data from lipids. I cannot tell you what the data are all about, but basically there are two conditions. And here, I mean, if you look at the picture, it seems like identical. But basically here we assume that under the two conditions, the network shares similarities, but they also have differences. That's why I have the two layouts, because here you see differences between the networks. And of course, then you need to look hard in this equation of what visualization tools to use in order to identify where the differences are and what it means from a biological perspective. And here, we have colored the different lipids classes appropriately so that you can see what happens. But basically, that's where we're going with this technology of how you can do this type of joint estimation of different correlation or partial correlation networks across different biological conditions, because that's where things get infinitely more interesting. Some, Charles Evans helped with some of this identification. Sasha Raskin with the data. Also, Steve Brown looked at some of this data with the SWAN data. And of course, we always need to thank MRC2 and to the grant that is paying us. Thank you very much. Chuck.
What's the magic ratio? So what's the magic ratio? So, as your, number of samples get below the number of features that you're looking at, you showed us that when you went to that plot that, when you went down to 100 and you had 1,000 features, you still were doing pretty good. But when do you start really dropping off? Or is this going to be a linear decline in the number? Because if you go to n equals 50, if you go to n to 25, when are you going to start gritting your teeth?
So, I mean, if you have 2,000 samples, 2,000 features and 100 samples, at most you're going to be able to identify a small fraction of the strongest edges. And my point is, don't even try. So that's where this is becoming key, and that's what we are working on right now. How to leverage additional targeted studies so that basically, it's not that easy to do because, it's not like, okay, I normalize all my data and then suddenly I go from 100 samples and 2,000 features to 500 samples and 1,000 features, because how are you gonna sort of align the features? But the key challenge is how you can sort of bring all the information that you have gleaned from all the other studies, and therefore you know that an edge between leucine and isoleucine is there 90% of the time, basically bring it in your estimation procedure. The technology, how you do the math, that's one part of my student's thesis. So technically I know how to do it, but how you process all these data sets in order sort of to create a background network that is sort of robust across a number of studies, that's what we're thinking, working, but this is the only way to go. I mean, if you're going to unleash this technology on 2,000 features and 50 samples, I don't bear any responsibility for what you're going to get out. It's not going to work. You need at least 300, 400 samples. If you're really trying to get a lot of stuff out. There is a hope with the lipids, because the lipids, you can do much more if you get into the lipid space. And the reason is that given the fact that you identify the classes, you can start putting in your estimation procedures many more constraints, because you know that the members of the lipid class are going to be much, much more highly correlated. And therefore, basically, you start narrowing the search space. As you can see, I mean, when you have fewer samples than features, you're trying to find this network living in an enormously complex and large space of networks. So the moment you start putting any interesting information, whether it is information about the lipid classes or chemical similarity, basically from a mathematical perspective, you start narrowing the search space that you're looking over, and that's when things start working. But 50 and 2,000 don't.
George?
Yeah.
George, I got a question from Gil. Let me read it to you. For many years, the peaks reliably identified in metabolomics assays seem to have stuck at a few hundred, maybe only 300 compounds. Are serious efforts being made by chemists to identify multiples of this number? Are known chemical modifications routinely detected? Aren't you worried about relying on co-expression with small number of confidently identified molecules to build networks?
I think this is a good question, but I think there are people in the room that can, I'm looking at Chuck or Alexi that probably can answer that question much better. I think it has to do with the quality of the data and, how confident you are about the input. And any thoughts? This goes beyond.
Well, there will be distraction to this.
You published on using variations of Granger causality to look at time series gene expression data. I was wondering if you have an update on the use of time series and how that might relate to this, and under what conditions might 100 repeated measurements yield better measures of edges and more valuable information than cross-sectional data?
So the moment, I mean, this is a good question. When you start bringing time course data, you have to deal with a whole bunch of other technical issues. And that's still work in progress. Because in that case, you need sort of to take into consideration because basically in time course data, you're going to have these thousands of features across different time points, and then you need sort of even to model the profiles carefully before you can start doing any of this. I mean, for any specific, because in that case, you have an even more challenging question to address. Eventually, we would like to go there, but there are a number of key technical challenges that still need to be thought out carefully.
So when we do sort of mapping various components, in our case genes, on a network with Yunfang Guam, their paradigm is basically that the network is something that she builds once based on the protein interaction profile, geodata, etc. And we are just putting some of our data on top. You're creating, if I understand you correctly, the network out of each experimental data set. And you hinted at the end that you might want to use the Metabolomics existing data as a background in the future. So how different is this? Is it that the correlation structure between metabolomics, let's say in blood or brain or some other sources, is very different? Is that why you have to reinvent the whole network every time, or how is this working?
No, I mean, basically, this is sort of the first instantiation of this technology. So obviously, you can start incorporating external information, what you know about connections between different metabolites from known pathways. So basically, I just basically showed you what you can do even if you start from scratch. But, in order sort of to start using it more routinely, as you hinted, basically you need sort of to start creating these background networks. But in this context, given the fact that you have all these unnamed metabolites, there are a lot of challenges of how you're going to integrate all your data sets. Because you don't, I mean, if I had identified all the metabolites, life is easy because I know what the columns of my data metrics are. But from study to study, I don't know whether this column corresponds to that column in the other data set. So you need to do a lot of additional work of how to get there and start integrating in order to create this common background networks. So it's a much more subtle game, but eventually that's where we want to go. But so basically what I presented was sort of version 1.0 of how you start with an appropriate data set, what are the technical challenges, and what are the key ideas that we are trying sort of to do, and with an eye on how this technology can be used either to clean your data set or to essentially identify your main features.
Sure. If I may.
Yeah.
So, I mean, this is absolutely true what you just said, but I just wanted to comment and put some numbers around it. So one key difference in sort of, doing this sort of mapping onto, essentially what you're talking about, I think, is sort of mapping your findings onto a knowledge-based network. Okay, and so that was the sort of, first and most obvious step, sort of borrowed from gene expression data, and so on and so forth, protein-protein interactions. And the trouble with that approach, specifically for metabolomics, is that the pathway databases, they tend to cover primary metabolism reasonably well, but beyond that, the coverage is very sparse. So it results in essentially, when you do this sort of mapping, in addition to what George said about the unknown features and so on and so forth, even for the known compounds, it results in coverage. Basically, you can map on average between 40 and 60% of metabolites, which is really sort of, given the already small numbers, makes it even more challenging. So I think it's sort of a must. What George described, seems like the way to go.
By the way, George, I have a question. So
Sure.
Maybe it's not relevant, but so let's say the blossom metrics, the blossom can you use the Blossom matrix, some sort of a prior or to get additional information? You ever thought about that? You know the Blossom matrix that is described in the protein field. So can you, I'm just saying that, have you ever thought about that? This is a protein is a protein involved here,
I mean,
right?
as long as, as I point out here, as long as you can bring any sort of prior information that you're comfortable with, the technical requirements, I can take care of that. So the question is, and that's what we have been discussing, and that will be something interesting to discuss with you, is where this external information that may be relevant in order to cut down on the search space is available. And after that, once you tell me that this is important and interesting information and relevant to what we're doing, I know how to take hand of that.
This is a statistical question, so it might be a bit boring for people. But in my experience, it's really hard to get this data to look multivariate normal. And so in practice, do these asymptotic results about confidence intervals and standard errors, are they really applicable? I mean, from the example that you showed, one way of interpreting it that there's a lot of false positives in it. So, yeah.
So, I mean, people have started working, and I have done some work of, if you have, so basically, the short answer is that as long as these deviations are relatively small, all these things go through. But if you really, really have a strong skewness and so forth, then essentially there is another sort of way of going through all this, basically through a regression framework. What I started describing of how you can get partial correlation from scratch without making the connection between the covariance and the inverse covariance metric. You can still get those sort of conditional dependencies, but the idea is that if you lose normality, you can still get your partial correlations, but there are some other technical issues that you need to be careful about. But in this case, unless I see, from a lot of data exploration that the assumption of normality is grossly violated, I feel fairly comfortable, especially when you concentrate on the strongest edges. The strongest edges are going to be recovered fairly well. When you start getting to this more gray area, depends on how many samples you have, then all these things get much more sensitive, and you need to be careful.
And a little bit
here, in principle, you have much bigger problems to begin with, given the lack of samples and the proliferation of features.
Maybe a little naive question from PGM point of view. So in the beginning of the talk, you were talking about like Bayesian networks, directly acyclic graph, and later in the end, in the algorithm and in the networks themselves, that look like Markov networks without directions. And they are equivalent only if you remove all the V structures. So if there are two metabolites that control the third metabolite, you always have a additional edge in between them. So is that what you're doing? Yeah.
Yeah. Basically, I'm looking at Markov network under the Gaussian assumption. But basically, the graphical structure that I'm looking at is a Markov network. Okay. Yeah. Sorry. Yeah.
Okay. Yeah. So in one of the table, you show the recovery by removing the subject samples, right? So have you tried to remove the features? For example, if some of the key drivers are missing, not in your data set, do you get a totally different network? Or but that can be a problem, right? If some of the key drivers are not in the data set, for example.
Yeah, because remember here, on the correlation network, this is not sort of going to matter because I'm looking at just marginal associations, or it's not going to matter that much. But on the partial correlation, if I start removing different features, the conditioning set becomes very, very different. And therefore, basically, the underlying structure becomes different.
So there's an approach called latent graphical model. They assume something missing, latent.
Yeah.
Okay.
So basically, yeah, if so in that case, basically, technically, what you work on, you don't work on sort of the covariance metrics and trying to do all this, but basically you work on the Schur complement of your covariance metrics appropriately defined. Then there are a whole bunch of issues of exactly how to do the screening if you start having such a framework. So there are a number of technical issues. If you do sort of vanilla latent graphical models, basically we work on the Schur complement of the covariance, that's fine. But here we first do this screening in order sort of to further cut down, and then a number of technical issues need to be thought carefully. That's a good idea, but as I said, this is version 1.0. All right.